Hello everyone, welcome back to another session of Systems Biology podcast. Massive bridges, massive dams, skyscraper, these are the marvels of modern engineering. All these huge man-made structures exist because our engineers can design them using the concept of mechanics and physics precisely. Now cells, a microscopic object, are also structures. Cells also have shape. And the structure of a cell and its shape is connected with its function. For example, when a cell divides, you can easily imagine a cell has to change its structure and shape because it has to divide into two parts. When a cell tries to move through, it has to reduce its rigidity so that it can move, so it changes its structure and shape again. Now the question is, can we extend the concept of mechanics and physics that we learn in our engineering classes to understand these microscopic objects, what we call cell, which are living organisms, living things? Exactly that's what my guest today is doing. Let us welcome Dr. Mandar. He is a civil engineer by training and in fact a faculty of civil engineering department at IIT Bombay. But he has not kept himself limited to civil engineering only. He has extended the ideas of mechanics, engineering, and physics to understand biology, particularly biomechanics, biophysics of cells and living organisms. So, welcome to Systems Biology Podcast, Mandar.
Thank you so much, Viplav, for having me here. Okay, really excited.
Okay, before I go into the discussion, I expect a very exciting discussion with Mandar today. Let me introduce the basic academic background of Dr. Mandar. He did his BTech in civil engineering from IIT Bombay. Then he moved to Caltech to do MS in civil engineering. Eventually, he moved to do PhD in doing applied mechanics in Caltech itself. And that's where he started interacting and moving into the interface of physics, mechanics, and biology. And eventually, he has a short stint of postdoctoral study at Brown University. And he returned back to India and joined IIT Bombay as a faculty. So let us roll the ball, Mandar. I will start straight on the basic question. You, over the years, have used the concepts of mechanics, physics, and engineering to understand cell and other biological systems. So, you giving some simple examples from your study or some other people's study. Can you make us understand that how these concepts of mechanics, engineering can be applied to understand living systems?
Okay, so that's a very nice question. So when I had joined, started my PhD at Caltech, so my supervisor, Professor Rob Phillips, so he was a world-renowned expert on material science, and he had written a really well-received textbook on that. And so I was planning to actually work on those areas, but when he joined Caltech, so he was at Brown University. So he told me that he has completely shifted gears and he plans to work in biophysics. So initially, even I was a bit skeptical because I had really no background at all in biology, and especially in my 12th class, like biology was really dreaded topic, and I had left many topics optional. Like I had no clue what those are. So I was a little bit skeptical, and I resisted working on those problems. But eventually, what I saw that there were many very interesting problems that were done by many of my co-group members, where some very basic ideas from mechanics and physics were implemented with a very interesting twist, and that kind of got me intrigued. And so the first problem that I worked on biophysics is on what are the forces. So, for example, we now know, many of us know viruses. So there are something called as bacterial viruses, the viruses which infect bacteria. And in fact, there is no no living cell, if I understand, for which there is no virus. Like for any cell, there will be some virus. So for bacteria, there are viruses. They are called as bacteriophages. And interestingly, what happens is that they have a head. So head is like a shell, empty shell, in which DNA, which is like a thread or a rope, it is really tightly packed inside. And my supervisor has this extreme knack of giving very evocative analogies. So the way he put it to us at that time is, imagine a FedEx truck. Okay, so in the US, there is a FedEx truck, it's a big truck, and you take out all the rope from the Golden Gate Bridge, and you can, and you pack it in that FedEx truck. So how crowded that is the same way your bacteriophage or a bacterial virus is packed with bacteria. Okay, and now this very basic problem, okay, looks almost like a mechanics problem. But then what happens is that the capsids, they have charges, DNA has charges, there are salts, there is osmotic pressure involved. So you have a basic mechanics problem, okay, which now becomes super interesting because there are all these additional features, and these are happening at very small scales. And when they happen at such small scales, many of you may be aware that thermal fluctuations, okay, they become extremely important. Okay, so you have this seemingly commonplace looping object, very small scale, where all interesting phenomena start to happen. And interestingly, what we showed in one of the works is that, and in collaboration with some of our colleagues there, is the amount of pressure, okay, that is built in a bacterial capsid, viral capsid, okay, if I remember correctly, is close to 50 atmospheres. So one could think of a viral capsid like a pressure vessel, so which is something which is very day to day. And then using some very, very simple ideas, you could find out that what will be the effective hoop stress that will be acting on the viral capsid, and can we think about what is the stability of the viral capsid, what does it depend on. And now these are the basic ideas, and now from these basic ideas you can go into various depths and make this problem extremely rich. And you can even ask, for example, that the bacterial virus, the way it infects bacteria is by ejecting its DNA into the virus. Okay, it doesn't physically enter inside. So there is a lot of pressure that is built inside the viral capsid. So does it use that pressure to actually kind of squirt out the DNA from the viral capsid? And so these are some of like ideas which are good parallels, okay, with what we see in microscopic world. But then it has its own specialities and peculiarities. Okay, so that's why these problems become very interesting, because it has a commonplace. And when I say commonplace, I don't mean that in a very pejorative manner. It's just like day-to-day objects and have a different twist on it, and the problems can become extremely, extremely interesting. And so that's what got me into biophysics. And then I continued working on various problems as a PhD student. I work on statistical mechanics. I work on partly on, for example, how is DNA packaged in histones. I did not take that problem much further, but I worked on that problem for some time. Then when I came back to India at IIT Bombay, I continued along those. And as I will discuss further, I also was lucky to find excellent collaborators working in various fields. And then I broadened this, my overall area of research. But the idea is that there are day-to-day objects. So you can think of various biological problems, like, for example, a pressure vessel. Or now, for example, I work in epithelial tissues. So epithelial tissues, you can think of them as, at a very simple level, as polygonal cells. They are connected to each other. Now, this can be modeled using what is called as there is something called as a vertex model, where now you can think of these epithelial tissues as a network of mechanical objects. And for example, you may have seen truss bridges, for example, on the roofs, okay, or for example, railway bridges. So you can think of this a cell network as some kind of an effective truss-like network, but it has very, very interesting properties, okay, which a standard truss network doesn't have. Okay, so for example, it can undergo turnover. There are active forces. There can be cell division. There can be self-healing. So it starts as a day-to-day engineering object, like one could think of that. But then there are many, many layers that are added to it, and that is one of the beauties of or challenges of applying these basic ideas to many biological problems that start interest. They start simple, and they can show extremely interesting behavior as you go deeper and deeper.
So if I ask to that point that the DNA packaging on histone, right? Many of my audience may not know clearly about that. Can you please briefly tell what is that?
Yeah. So this is another point. So I do not actively work on these problems, but for example, I have had some collaborations in the past. So what happens is that for example, we know that if you just stretch out the DNA. Now my advisor had written this book on biology by numbers, and there I think there is a very evocative analogy. I do not know. Uh, uh, I don't want to put some wrong analogies, but the DNA is so long, okay, that if you want to just, for example, take that DNA and put it inside the cell is too short in order to pack that DNA. So various
Yeah.
hierarchical,
So
yeah,
you mean the length of the
the length of
DNA
the
is so
DNA is so
big
big.
that I cannot pack it so easily in the cell?
Or let me put it this way. So, for example, in polymers, there is this very simple idea. Like when I say simple, what it means is that it's a well-known idea that a simple polymer, okay, of a length L, if you ask it what is the radius, so when you put a polymer, it kind of goes into becomes something like a ball. So if you ask that if the L is the length of the polymer, then the radius of the polymer, okay, it approximately goes as L to the power half. Okay, so this is like one of these what is called as a Gaussian model. Okay, and if you use that simple model, then the size of the DNA ball that you get is significantly larger. Okay, compared to the where L is the length, where compared to the size of the cell. So you need some mechanisms in order to package it nicely. And there are these various mechanisms. For example, histones are, for example, are these proteins, are these structures which are made of like a group of four proteins, I think H1, H2, H3, and some H4. So you have those proteins. Then DNA, for example, there are various chaperones and there are various energetic considerations because of which DNA, which is a thread, it gets wrapped around them, and then that is the first primary structure. Then those, for example, get kind of undergo further interactions. They get packaged further and further until, for example, you get this so-called chromosome-like structures which we see in textbooks. So there are a various hierarchy, okay, by which this uh, uh, huge DNA length, it gets ultimately packaged into a tiny or relatively tiny length, which ultimately can be packaged in a nucleus. Okay, so I may not be fully correct exactly in what I say, but this is the general idea.
So in a way, it's the engineering problem.
It is
How can you pack a small, large thing into a small one?
correct, correct. So it essentially becomes an engineering problem, but where it becomes exceedingly like where it kind of really departs from being a standard engineering problems is there are various active mechanisms. For example, in order to really do this packaging, because another thing we have to understand is that the cell doesn't have infinite time to do all these things, so it has to do it quickly. So, for example, there are many active processes which, for example, will make sure, okay, that these things happen fast. On top of that, it is very specific. For example, there are DNA, for example, has various chains, okay, various regions. We call them as genes. Okay, again, I'm not an expert, but genes will express proteins. So at various cells, at various times, you need them to be exposed. Okay, so they need to be bound in a particular way where they can be accessed at later times. So there are various constraints. Okay, so entire biology, okay, it lives under constraints, and those constraints are of space and time. Okay, and timing. And so all these things have to happen, okay, in with respect to those. And here I'm not even mentioning that all these things have undergone billions of years of evolution. So they are all operating under this backdrop of evolution. So these so many constraints are there. So a standard engineering material will kind of, okay, you create the analogy, you use certain ideas, but when you go to specifics, then they will not directly be applicable for these problems. So that is a key point.
Yeah, I think there that what you are hinting is that the limitations. If I just want to make one-to-one projection of engineering concept to biological problem, I'll come back to that issue.
Yeah, but
Before that,
yeah, please.
I want to pick up one particular interesting work that I was reading through your papers, and what I saw very recently, you did a collaborative work with experimental biologists where you try to understand the biomechanics of E. coli cell shape, right?
Yeah, yeah.
So that paper I was reading has both experiments as well as mathematical models to understand the biology, right? So can you please tell us briefly what was the problem and how the biomechanics and the mathematical models based on biomechanics helped to understand the biology better in that particular.
Yeah, sure. So this work was done in Professor Chaitanya Atle's group, who is at IISER Pune. So when Chaitanya was discussing this problem with me, so he already had a brilliant experimentalist, Tanvi, and he also had a student, Rith, who was working on modeling. But the starting point was a pretty simple question: that how does E. coli maintain a stable width, and what controls and if you do any kind of genetic perturbations which for example change expressions of any structural proteins, okay, which for example build the walls of E. coli or it creates a structural framework, then it collapses the standard cylindrical shape. It collapses into, or it becomes a bulge shape. Okay, so the point was that is there some kind of an overarching idea by which we can understand how this transition happened. So my kind of role was to step back from all the molecular details and to frame this as an effective mechanics problem, and where, okay, like in really joint collaboration, and I was mostly advisory. So we worked on, for example, treating the cell envelope like a parametric shell. So when I say a shell, for example, shell is essentially nothing but a structure with a very thin wall. For example, you can think of,
Maybe mesh or made up of mesh or something.
it is made of a mesh, but you don't necessarily need to think about it. For example, you can think of a dome, for example, or even pressure vessel. Pressure vessel is also a shell. So the cell envelope was then modeled like a pressurized shell, and it had bending rigidity and surface tension. Okay, and then we worked out these equations. And what was really interesting was that the model predicted that there is a particular value of internal pressure that appears, okay, which we can do experimentation and falsify it. And so by adjusting the external osmotic pressure, what we could see is that the bulging could actually reverse, and whatever was the prediction of this model, okay, it could actually be verified. So this, for example, is one interesting example among so many others, where what mechanics does is it doesn't necessarily go into each and every microscopic details, but what it does, it provides some kind of constraints, and your internal biological process, what they do is that they are subject to those constraints, and you can use effective descriptions in order to see how an interaction between real effective biological processes and mechanics together, okay, gives rise to an overall phenomena. For example, in this case, which is the shape of the cells.
Yeah, this brings me to a very common question that usually not just biologists, even any other people from engineering background also many a time think of, that how can you imagine that you will use a simple mechanics based or a simple physics based model to understand a biological phenomena which require lots of processes, hundreds of molecule, all these things are involved, right? So what I understand just now, as you said, that when you are building that mechanics based model for the shell of the E. coli cell wall, you are actually removing the bells and whistles and focusing on the some elementary thing, right? So how do you look at this criticism that whenever people like you create these type of models, like even many physicists and engineers who try to build model for biological system, that they usually get rid of the detail and focus on certain aspect only and try to create the model?
Yeah, that's a very nice question, and this is something which is I do not have a 100% correct answer, and I don't think anybody has a 100% correct answer. But there was earlier, like say maybe say 50 years ago, or maybe 50 is a bit too much, but maybe around 30 years ago or so, when computers started to become more and more powerful, then even, for example, in material science, various scientists, for example, started to do full-scale molecular dynamics simulations. Previously, for example, mechanics, continuum mechanics ideas, okay, which doesn't take into account all these molecular details. The molecular details only show up as effective mechanical properties. So many scientists, they did molecular dynamics simulation in order to predict various features. And the belief was that if you go to smaller and smaller structures, then at some stage continuum mechanics will break down. Okay, but interestingly, it turned out that even at the scale of a few atoms, okay, continuum mechanics ideas, they work surprisingly very well. Okay, so what it means is that if you are interested in broader phenomena, which, for example, in technical language, what are called as hydrodynamic phenomena, or large wavelength and slow timescale phenomena, okay, then what happens is that many of these molecular details, they are not particularly important, because, for example, they happen fast. Their effective deformations, what they create, okay, on the scales that you are interested in, will only be explainable or only be understandable from a more continuum or from a more so-called hydrodynamic point of view. So there, for example, depending on what scale you are looking at. So for example, if you really want to understand how are the microscopic reactions happening, then we may not be able to use the model. But if our overall goal is to know, for example, what is the shape of, say, E. coli, or for example, how is my tissue deforming, on what is the scales over which the tissue is deforming, will it undergo a fluid-like transition, will it undergo show a solid-like behavior. So those are problems which happen on scales, time scales which are slower compared to many molecular time scales, on length scales which are much larger compared to many molecular length scales. And on those scales, magically, we can forget about these details. And in fact, it is better to forget all those details because they will create a lot of noise. And when you do that, then almost magically things start giving you details, and you can understand the phenomena on those scales much better using these ideas.
So essentially what we are saying, depending upon the question, sometime I have to forget about details of each tree and focus on the forest as a whole so that I can understand certain phenomena,
Yes,
which will be difficult to understand if I focus on each of the tree there.
that is very correct. Okay. In fact, not only in this case is it detrimental to look at all the tiny details. Okay. So they will not help you much because they will just create, like you said, that you will mistake the forest for trees. Okay. So that's what will happen. So here, continuum ideas, they work brilliantly well. And one may argue that what is the meaning of continuum? So the meaning of continuum just depends on the scale. For example, at the scale of cell, your intracellular fibers and filaments, you can think of them as microscopic, overall cell as continuum. Then when you go to the level of tissues, then you can think of cells as your microscopic elements and your collection of cells as a more continuum. When you go on even bigger scale of organs, then you can think of, for example, tissues to be your microscopic elements and your bigger organs, okay, as continuum elements. So there is always a hierarchy, and it just depends on where in the hierarchy you are, and based on that you decide what is the level of modeling that you have to do. And at every level you have certain curious phenomena, especially in biology, that will keep appearing.
Yeah, this brings me to another question come to my mind right now. See, I started this dialogue today by talking about cell, but interestingly, you brought the idea of tissues and higher things. And for tissues, you said that you use one type of modeling approach called vertex models,
Yes.
right? So, okay, can you tell us that what is that in a bit more detail? What how you model a tissue? If I understand, the tissue is nothing but a collection of or ensemble of cells connected with each other, mostly same type of cell. So epithelial will be a layer of cells, right? What is that vertex model and how you actually use that to understand this layer of cells?
Okay. So vertex model is has been around for actually quite some time. Okay, almost from early 80s, and there is some pioneering work by George Oster where he used this vertex model. But I think his modeling was way ahead of the time. There were no experiments, or the technology was not sophisticated in order to really figure out or predict or to verify what his models predicted. So these models have been around for a very long time. Now, what these models are, in very simple words, is epithelial tissues. They are connected group of cells. Now, when cells come together, these epithelial cells, they form a polygonal network. Now, the cells, epithelial cells, typically have a top layer and a bottom layer, called as an apical and a basal layer, and they are connected to each other by what is called as a lateral layer. Now, in many phenomena, for example, think of that as a Drosophila. Drosophila is a fly. So during its wing development, okay, there is a lot of development that happens, for example, almost in 2D. Okay, so there are many other intermediate features where 3D features are involved, but there is a particular phase of development. In a lot of development actually happens on planar geometries, and when that happens, what you can idealize is you can say that I do not really want to worry about my top, bottom, and the thickness, but instead I will just look at the top surface or the so-called apical surface, and this surface is essentially nothing but now a group of polygons geometrically, and each polygon is made of vertices, and the vertices are now degrees of freedom. What do I mean by that? That if I want to evolve the tissue mechanically, then the nodes or the vertices move, and the movement of the vertices changes the geometry of the tissue. So this is the general idea behind the vertex model. So geometrically, it is a collection of polygons represented by vertices as degrees of freedom, And now what you do is that you imbibe mechanical properties on it. You say that there is a cortex which is made of contractile actomycin. Cells like to adhere to each other, so there is some effective surface tension, okay, or effective adhesivity. Then the cells, for example, don't want to be squeezed because they have a particular volume. So that gives rise to internal volume-like forces. Now, cells not only can do that, they can also divide internally. So then you can have internal division on this. Okay, so that will create another level of forcing. Moreover, you can argue now that cells are these tissues are these active objects, and they have internal cues which tell them in what direction to deform, in what direction to divide. So they have what are called as planar cell polarity or PCP systems. So you put some kind of a polarity systems. Now the cells can develop lamellipodia in various cases. Okay, lamellipodia are these structures that they develop in order to move. So then you can then represent those effect of lamellipodia as motile forces. Okay, now this is what I'm discussing, like the overall general geometry, mechanics, internal forcing. Moreover, one of the most important properties is remodeling. That the cells can not only, for example, divide, they can die, okay, and can be extruded out. And they can also, for example, undergo neighbor exchanges. Now, this remodeling and active forcing and overall mechanics, what it does is that it gives an incredible rich mechanical behavior. Forget about biology. Even from a mechanics point of view, it gives an incredibly rich mechanical behavior. So much so that, for example, by tuning various of these properties, the cells can, the tissues using this kind of a model, and also in reality, can exhibit rheology, which on the one end can be like purely solid like, and on the other end it can be extremely fluid like, and in between it can behave like complex fluids and complex solids. And now actually in reality we see that in Drosophila, which is fruit fly, or in zebrafish, all these processes, they do come together, and ultimately what they do is all these internal processes, some kind of signaling, some kind of molecular scale processes are happening. But effectively at the final scale, okay, what is called as a downstream scale, forces are generated, remodeling happens, movements happen, and ultimately you sculpt the tissue. And this is where mechanics happens. Okay, and the vertex model, for example, is one way, in order, in a very simple way, you can depict this really rich mechanics.
So as I get it, the first thing is you have done that, you have removed the details. You focused because you simplified it, because it is workable. It is a 2D geometry. You converted that. And then going into details of each cell, what it is made up of, you have essentially considered them as a polygon connected with each other. And then if I forget for the time being about this tissue remodeling and birth and death of those things, those make things a bit complicated. What you are, apart from that, what you are doing now, you have a mesh of polygons, two-dimensional,
Yes.
and you are using some sort of force, stress, strain, all this type of thing, right? And you are allowing the mesh to evolve with time,
Yes.
or this depending upon the forces, right? That's what you are doing.
Effectively, yes.
And then you are putting additional thing.
Yes.
For example, you are saying that there are maybe active processes, as you are repeatedly saying, right?
Yes.
And there may be a neighbor exchange, or death of a cell, a new polygon originate because a cell divide. All these complexities will come, right? Am I correct?
Yes, so exactly. So yeah, so it is like there is a vanilla vertex model, and on the vanilla vertex model, now depending on what level of details you want to add based on the problem that you are addressing, you can increase its complexity to whatever level you want to.
Yes, so this brings me to one important question. See, what we are doing is we are in a way simplifying, making ourselves comfortable, so that the system that we are dealing with now amenable with the concepts of physics and engineering that we learn actually in the college, right? Now, but as you are working in this field for long, do you think that the physics at this microscopic level, right, starting from mechanics to rheology to other aspect, because you are repeatedly saying that active process or a cell is an active thing. So if I understand that you consider those bridges or a ball rolling down a hill, those are actually passive things, right? The standard physics and engineering thing that we learn in school and college are actually passive object, where you consider you, me, and a cell as a active object, and they are doing active processes. So when you deal with these active systems, active agents, whatever you call it, and at that microscopic level, eventually, do you think somehow our standard physics and engineering concept may break, or we need to develop some new physics and engineering concepts there?
So that's a very good question. So the key word for biological systems is essentially that they are not in thermodynamic equilibrium. So that is a key word. So for example, most of the non-living objects that we look at, or the man-made structures, human-made structures we look at, they their thermodynamics is straightforward. For example, if you build a building, okay, it is not suddenly going to start generating internal forces. It may, because for example there is some corrosion that may have happened, so that could be a change that can happen. Or for example, if you expose it to elements, some parts may expand and start putting in forces. But those are not active in the sense that what do I mean by active is that uh, there is no simple way to think about it, but they are always, there is some source of energy that is being given to these biological systems. Okay, and they, in many of them, they remain in what is called as a steady state. So what do I mean by steady state is, if you look at it, at the system, then on those observational scales the system is supposedly not changing, but there is always some level of flux of energy and material that is happening to the system. So this is one of the big differences between biological systems and any standard engineering systems. So that is not to say that standard engineering systems cannot be made active. In fact, there are many researchers, for example, they have systems which are not technically living systems, but they show various interesting behavior similar to living systems, because they have interactions and energy influx which is not exactly similar, but which is akin to what we see in various biological systems. So does that partly answer your question?
Yeah, yeah, to some extent, definitely.
Yeah.
So there is obviously that, as you said that, for example, I am at a steady state right now.
Yeah.
My body mass in general, shape, size is not changing by day-to-day basis in small time scale.
Yeah.
But to maintain this body, I have to continuously consume food so that I generate energy, so I can repair my body. So anyway, my whole body is a active system,
Correct.
has active processes. I am not passive. If I become passive, obviously I will not be able to hold this shape and structure. But my shape and structure is in a steady state.
Yes.
Right.
Yeah.
So this is quite different from a chemical reaction happening in a test tube where things become eventually equilibrated. Or as you tell, the building I have constructed, this is in equilibrium as a whole. All of it is not continuously breaking down and making itself continuously.
Correct.
Right. If it breaks down, I have to do it out from outside.
Correct.
It is not itself will do
Correct.
that. Right?
Correct.
So does that falls into this idea of active passive, right?
Correct. So broadly speaking, so there can be some subtleties in that, but overall, I think I agree. This is exactly that. And in fact, even if I'm just sitting, not doing anything, okay, so supposedly for an observer, I'm not doing anything, but internally the body is working furiously. There is continuously blood circulation happening, motors are working, the muscles are working constantly to just make sure that I don't fall down. So even supposedly externally nothing happens, internally there is some kind of a churn that is always happening.
Right. Yeah, and I think although most of the time when these examples comes to our mind about ourselves, but I believe a single E. coli is also doing that continuously.
Correct. It is continuously doing that. There is some level of its genome, for example, or its DNA. Okay, it is processing it. Then there are some proteins which are constantly repairing the DNA, and cell division is happening. Activities are happening to figure out when the cell division will happen. So even a tiny DNA, even a tiny bacteria, will, for example, is always in a state of churn, and it is not in thermal equilibrium.
So if you allow me to extend the same question, see, is there some from your experience or some of your colleagues or other people's experience, is there any material properties or physical properties that usually we do not see in our bulk material or engineered systems that you can see at cellular level? Is there any such thing like that, for which maybe we have to go for a different types of physics or something?
So now the question is not a different kind of physics, but it seems like that you have to repurpose the existing physics and have possibly better ideas for modeling non-equilibrium thermodynamics, because non-equilibrium thermodynamics is a really an open topic, as I understand, that what we kind of really understand is standard thermodynamics. We also decently understand thermodynamics close to equilibrium, but thermodynamics really far from thermal equilibrium is something which is not at all. It's completely an open area, and a lot of people, even in physics, are working on it. Not just biophysics, in physics are working on it, and the belief is that these ideas from non-equilibrium physics, okay, are in principle sufficient to understand biological systems, albeit with some level of repurposing. So that is the point. So I don't think, uh, uh, or I may be bold in saying that, but it doesn't seem to me uh, that any new physics is required or any new ideas are required in order to understand biological systems. But possibly we need better development in understanding of non-equilibrium thermodynamics, is what I can say.
Yeah, that's interesting because always what I have found is that when we learn different concepts in physics, they are usually has a limited domain for it, right? So when you talk about atomic structure, it is you are working at the atomic level. When you talk of gravity, you are largely talking about large object, right? But thermodynamics, I always have found something which is usually don't bother about the system at what level you are working, and you try to use thermodynamics concept as a truth everywhere,
Yeah.
right? So as you said that the classical thermodynamics possibly is not capable to handle all these issues,
Yes.
and that's why everybody is trying to now develop this non-equilibrium thermodynamic concepts. And I think there should be some, as more people are working, new breakthroughs will be possibly coming in that.
Yeah, most definitely. And there is another point which I did not answer. I forgot to answer is what are the specific behavior that, for example, these systems show, which your passive objects cannot exhibit. So one clear-cut phenomena is, for example, just general modularity. For example, if you look at a single cell, okay, so for example, a single cell it can be just static, just is on a substrate, and it is not doing much. It is just randomly extending or fluctuating around. But for example, if there is a food source or if there is a chemical signal, then there is a huge amount of internal modifications that happen. For example, you have a fiber-like network of actin which can break down, and it can undergo, for example, a gel to sol, okay, a gel to a solution-like transition, and then start migrating. So this is something, okay, and this happens like pretty much spontaneously, that you do not have to go and instruct the cell. The cell exactly knows that this is happening, and it will start migrating. So this level of modularity, changing its material properties, okay, pretty much on the fly, undergoing fluxes and flows based on what are the external cues, and for example undergoing, like in a collective, for example in a tissue, if you for example create a breach in the tissue, then figuring out ways to undergo wound healing, okay, where it will close the gap. Or for example, if you cut off one signal, then there is some internal another network will, for example, will become active, and you will have a different mechanism by which, for example, the cells can migrate or the cells can divide. So there is innumerable ways in which the cells can just change its behavior, mechanical behavior, and which is something which you would not see in standard passive materials. So this is really, really the hallmark of living materials, and maybe some metamaterials or some active metamaterials that people are slowly developing, but they are nowhere near the scale at which living materials show this adaptability.
Yeah, that's very interesting. In fact, you mentioned metamaterial because I was thinking that it is somebody from material science background or engineering background will also try to incorporate this ability to morph from one property to another property. So my car can morph into something else, or even a simple material that I am using right now, right? So that may morph and change its mechanical properties. So depending upon the external cues that comes to it, right? That will be great, right? So if we can learn from biology and then use them in material science, that would be great.
So there,
And as you said, that some people are working
so
on
there are,
that,
like,
right?
I do not follow this literature so keenly, but I have seen that there is this entire area what is called as biomimetics, where what you do is you look at various biological structures, various biological processes, and then you try to look at what is the utility that is served by those processes, and you try to mimic the features so that you can have artificial structures which can also show similar behavior. And that's a huge field,
Right, right.
and there are many developments that are happening, but I unfortunately don't keep so much track of that. But that is another really interesting
Yeah,
area.
there are lots to actually to consume. But in these 30 minutes of discussion, we already have learned lots so many keywords. So, is there? Can you suggest us some sort of some popular science books where people can read? After the dinner book type thing, which people can read to understand this biophysics and biomechanics aspect of cell and biological systems.
What I can do, so rather than suggesting direct titles, okay, so uh, I can just suggest an overall idea. Like for example, one can just go on. There are so many books, and there are various levels. For example, there are books on, for example, understanding DNA. Okay, so if you are somebody is
Okay.
like somebody comes from a little bit of a physics background with interest in polymers, then there are popular books on DNA mechanics. Okay, just do a simple search on Amazon, you will find those books. Then somebody who is more involved in chemistry aspects of biological systems. Then there are books, for example, more on these systems and chemical reaction like aspects. Somebody, for example, who has a more mechanicsy flavor. So there are books on, for example, understanding biology or understanding general soft matter from mechanics point of view. And somebody who, for example, wants to generally understand how organs form or how developmental biology happen, which is morphology, morphogenesis. There are books, many books on uh, those topics also. So what I could do is that maybe afterwards I can send you a list. If and if you are, for example, you
That
can
will be
put it at
great.
the
That will be
bottom
great.
of it that these are the
Yeah,
references,
yeah, I
and
can
somebody
put it in the
can
video
put that.
itself somewhere.
Yeah, and I will
Yeah,
also
that will be
advertise.
great.
Yeah, and I will also advertise that with some of my colleagues, Vijay Kumar from ICTS and Gautam Menon from Ashoka University, and Pramod Pularkar from Raman Research Institute. We very recently wrote a review on, like we have been working on it for past many years, on forces in biological systems broadly, and the paper is on archive.
Yeah, I was reading
Yeah, yeah.
that.
So
It's a very
yeah. So
interesting.
that could also be one of a good starting point for somebody at maybe senior undergrad or early grad level to understand various of these aspects, because many things that we discuss
Yeah, yeah.
today, they are there in much more detailed format
Yeah, I
in that.
saw. It's
Yeah.
very nicely written. In one place, everything is almost
Yeah.
there.
Thanks.
Yeah. So I will put that in the video also so that
Sure.
people can download and read it.
Sure.
It is an archive, so anybody can download and read that
Yes.
easily.
Yes.
This brings to me a student and undergraduate related issues and postgraduate PhD student related issues. So as I was looking into your publication profile and the people you have worked with, you worked with a spectrum of people who are theoreticians, physicists, engineer, biologists doing hardcore experimental biology, right?
Yes.
Uh, can you tell us one thing? See. When you work with students or even co-supervise people from different backgrounds also, right? So when you try to work with students from different background, right, and in a group, what academic credentials or capabilities that you look into those students?
Yeah. I think the biggest credential is just curiosity, wanting to work on things, and having the agency to do, like, learn various things as and when they are needed, and march forward. And really, these are really all the skills that are needed nowadays, because especially like in the last 1 year, and more so like in the last few months, you look at various of these LLMs and AIs, like you have Gemini, you have ChatGPT, Grok, Claude. The barrier for understanding any new thing is almost zero. So previously, for example, you could like, there is a big paper, like you see a big paper with lot of jargon, and you want to understand it, or there is an article, there is some blog. It was very difficult. Like you would need to find some person and ask them to explain. Now you just feed that to these LLMs, and you would pretty much get, if not the expert level advice, a very good expert level advice. So if you just want to do things, and you are like want willing to learn and put efforts. So I think that's all the quality that I really care about in anybody who wants to work.
So, connecting to the same question,
Yeah.
because I get this question many a times through emails and other posts, that nowadays many students from engineering, physics, math, right? They are excited because there are lots of biology related popular science books they read, and in media also they read about lots of things happening in the world of biomedical sciences. So they are excited, but many a time they feel that they are constrained, that they do not know the biology, so there are apprehensions that they whether they should look into and jump into the field of biology and biology research or not. So as you said rightly, that nowadays information at least has got democratized. So if you have an internet connection, if you can dig into these papers, you can use chat gpt or something like that to make things simple. Hundreds of YouTube videos are there to understand those, right? So learning is no more a barrier if somebody want to study, right? If you want to learn that, but beyond that, you yourself was not a engineer biology student, as you said earlier. Actually, you moved into this whole field during your PhD. Your PhD supervisor has a obviously some lots of contribution in that directions also. So if somebody is doing right now undergraduate or first year, second year engineering student, so what will be your suggestions if they want to explore this world and test the water?
So I think the one of the key ideas is from for example, like as you would see from my example, that I did not explicitly plan things. So I think serendipity is one of the really key features in life. You start doing X and you never know when you get pulled into Y, and it happens for the best. So one at some level and is just be willing to do things, be open all the time, keep your eyes open, and something will happen. It is not at all clear that if it will be biology or if it will be something else. But the interesting thing about biology is that it is at the interface of life sciences, engineering sciences, mathematics, physical sciences, chemistry, and at the interface of computer science, electrical engineering, whatever you think of. Okay, there will be some problem in biology which can be thought of in that particular way. So that's why, for example, you may have any background, but very likely there will be some or the other problem in biology, which, for example, will be amenable to whatever expertise any student can have. And nowadays, I think all the watertight compartmentalization is kind of just not relevant. That you work into one area and it suddenly morphs into something else. So you can always start with something that uh, you are familiar with. The barrier to learning is very low. So you can kind of start with one and slowly morph into wherever the problem takes you. And how exactly will it work out is, I think, anybody's guess. And I really myself do not know what I will be doing like maybe 2 years from now. So, but only thing what I know is that you keep working. Uh, you start with something you know, and keep following as per your understanding and your taste, and something good will definitely come. There is no question about it.
So I like this too. You actually hammered the two critical things. That one is obviously curiosity, right? And the second one is to keep be open-minded to, as you go to get opportunities and in your journey, to grab those opportunity and try the water. You never know whether you will be successful or not, but if you keep the sail at the you don't take the boat to the sea, you will never know whether you can row the
And the
boat,
key
right?
point is
So the
that
same
you
thing
may
here.
start to do X, but you may do Y and end up doing Y very very well. So that is thing which I have seen again and again.
Yeah.
Yeah.
Yeah, that's very encouraging, because many a time I see youngsters have apprehensions, obviously, because their apprehension whether they will be able to cope up, whether to learn that subject or not. But as you said, and I also see lots of people coming from different backgrounds, eventually becoming very successful as a biologist, as successful as a trained
Correct.
biologist,
Correct.
actually.
Correct.
And the last thing is essentially, as I personally also have felt, I think you have surely felt, you do it because it is fun. You enjoy doing that.
Yeah.
That's the whole point.
Yeah. So the point again is this, that even I tell my students also various times that you do it because it's a lot of fun, and because it's a lot of fun, whenever there are periods when it is no longer fun, okay, like the fact that you have tasted that joy, okay, it sustains you many times.
Yeah,
Because there
that's
are
right.
inevitable
So,
periods that will happen, like any time in research,
yeah,
that you keep
obviously.
doing
Yeah.
something, nothing works. But since you have tasted the joy, okay, it keeps you afloat in these cases.
Yeah. So again, if I take the example of this active process or active systems, we human beings are not passive. So we are active systems. So we continuously, get the energy from our successes. So when we are in a downtime, those successes and in fun that we have in the good time, they actually pushes us to keep our journey. And as you said, we have to be open-minded. So that means just like other active material, we have to change our shape, structure, material property to learn new things,
Yeah, yeah.
right?
I think being,
So that's a,
yeah, I agree with you. So being fluid in your approach, I think is a good skill.
yeah.
Yes.
So not to get compartmentalized in one particular area and like that.
Yeah.
Yeah, so Mother, thanks a lot. We have lots to chew in this particular discussion. We started from a single cell, and what is very interesting to know that how you trained as a civil engineer and moved into this interface where you are still a civil engineer faculty and you still teach structural engineering to your student, but at the same time you are interacting and working with people from other background and using the concepts of physics and engineering to understand life as a whole. Because I believe at the end of the day, every day when I see myself in the mirror, I find that okay, we know so many things about the universe. We can create such a massive structures, massive things. We are thinking about going to Mars and building a civilization there, but we know so little about
Correct.
ourselves. Biology at the end of the day is learning about ourselves, where we come from, whether I'm an experimental biologist or like you coming from engineering background. So thanks a lot for sharing your thoughts and about the different ideas that you are working on. I'm sure my audience will be also enjoying will also enjoy this discussion and find this very fruitful to explore new areas, particularly biomechanics, biophysics, the way you are doing right now. Thank you, Mandar.
Thank you so much for inviting me.